Carbon dioxide sits at the center of the modern climate conversation, usually framed as a problem to be reduced, captured, or eliminated. Yet from a biological standpoint, CO₂ is not a pollutant in the traditional sense. It is a foundational input to photosynthesis, the chemical process that makes nearly all life on land possible. Plants do not merely tolerate carbon dioxide, they require it. Without it, terrestrial ecosystems collapse. This simple fact creates a tension that rarely survives the compression of headlines. To understand whether rising atmospheric CO₂ is making vegetation grow more easily across the globe, we have to step away from slogans and return to first principles. Photosynthesis operates by taking carbon dioxide from the air, water from the soil, and energy from sunlight, converting them into sugars that fuel plant growth. Under controlled laboratory conditions, increasing CO₂ concentrations almost always increase the rate of photosynthesis, particularly in so-called C3 plants, which include most trees, wheat, rice, soybeans, and nearly all temperate vegetation. This phenomenon is known as the CO₂ fertilization effect, and it has been experimentally observed for more than a century. What has changed over the last 150 years is the background concentration of CO₂ itself. Prior to industrialization, atmospheric levels hovered around 280 parts per million. Today, they exceed 420 parts per million, an increase large enough to matter biologically. In growth chambers and greenhouse experiments, this shift alone often produces measurable increases in leaf area, biomass, and water use efficiency. Plants tend to lose less water through their stomata when CO₂ is abundant, meaning they can grow more with the same, or even less, moisture. The critical question is whether these controlled results scale up to the real planet, where plants are constrained not only by carbon, but by nutrients, temperature, water availability, soil structure, pests, and competition. This is where public understanding often becomes distorted. Media narratives frequently imply that any biological benefit of CO₂ is either negligible or irrelevant, while skeptics sometimes imply it is universally beneficial. Neither position survives close inspection. Satellite observations over the last several decades have revealed a measurable and widespread greening trend across much of the Earth's land surface. Large portions of Eurasia, Australia, parts of Africa, and even arid regions show increases in leaf area index and vegetation cover. Multiple independent datasets, using different instruments, methods, and research teams, converge on the same broad finding. The planet has, on average, become greener since the late 20th century. This is not speculation, it is direct observation. Attributing that greening to a single cause, however, is far more complex. Rising CO₂ plays a significant role, but it does not act alone. Nitrogen deposition from industrial activity, land use changes, irrigation, crop breeding, and climate shifts all interact. In some regions, warming temperatures extend growing seasons, while in others heat stress or drought counteracts fertilization benefits. The Earth is not responding uniformly, it is responding unevenly, region by region, ecosystem by ecosystem. This complexity is often flattened in public discourse. Greening is sometimes dismissed as a distraction, while damage is emphasized as a certainty. But the scientific literature does not describe a world that is simply getting worse everywhere, nor does it describe a world being saved by carbon dioxide. Instead, it describes a biosphere under changing boundary conditions, adapting in some places, struggling in others, and responding in ways that are measurable but not easily summarized. What matters for this series is not whether CO₂ is good or bad, but whether public perception accurately reflects what researchers actually observe. Early evidence suggests a gap between the two. Rising CO₂ is not merely an abstract climate signal. It is an active biological input, one that alters plant physiology in predictable ways. Ignoring that fact does not make it disappear. It only makes our understanding less complete. In the next segment, we will move deeper into the mechanics of plant physiology itself, examining why some plants benefit strongly from rising CO₂ while others respond weakly, and how water stress, nutrient limits, and photosynthetic pathways determine who gains and who does not in a higher carbon world. To understand why rising atmospheric CO₂ produces strong growth responses in some ecosystems and only muted effects in others, we have to move inside the leaf itself. At the microscopic level, plant growth is governed less by ideology and more by biochemistry, specifically by how plants capture carbon and manage water under imperfect conditions. Most of the world's plant species use what is known as the C3 photosynthetic pathway. In these plants, carbon dioxide enters the leaf through small openings called stomata and is fixed directly into a 3-carbon molecule by an enzyme known as RuBisCO. This enzyme is ancient, inefficient, and central to the story. RuBisCO does not only bind carbon dioxide, it also binds oxygen. When oxygen is captured instead of CO₂, the plant initiates a process called photorespiration, which consumes energy without producing sugar. From the plant's perspective, this is wasted effort. Higher atmospheric CO₂ concentrations tilt this competition in favor of carbon. With more CO₂ available in the air, RuBisCO is more likely to bind carbon rather than oxygen, reducing photorespiration and increasing net photosynthetic efficiency. This is why C3 plants, trees, shrubs, most crops, and nearly all temperate vegetation tend to respond strongly to CO₂ enrichment. The biochemical bottleneck that has constrained them for millions of years loosens slightly, and growth becomes easier, not because the plant has changed, but because the environment has. A smaller but ecologically important group of plants follows a different strategy. These C4 plants, which include maize, sorghum, sugarcane, and many tropical grasses, evolved a workaround to RuBisCO's inefficiency. They concentrate CO₂ internally before it reaches the enzyme, effectively eliminating photorespiration even at low atmospheric CO₂ levels. This adaptation allows C4 plants to thrive in hot, dry, and low CO₂ environments, but also means they gain relatively little from rising atmospheric CO₂. Their photosynthetic machinery was already optimized for scarcity. This distinction matters enormously at the planetary scale. Forests, woodlands, and high-latitude ecosystems are dominated by C3 plants and thus show strong greening responses. Many grasslands and savannas, particularly in warmer regions, are dominated by C4 species and show weaker or more complex responses. The global greening signal is therefore not evenly distributed, it is patterned, and those patterns align closely with plant physiology. Water use adds another layer to the picture. When CO₂ concentrations rise, plants do not need to open their stomata as widely or as frequently to acquire the carbon they need. This reduces water loss through transpiration. In effect, higher CO₂ allows many plants to grow more while using less water per unit of biomass. In semi-arid regions, this improvement in water use efficiency can be as important as the fertilization effect itself. It helps explain why greening trends have been observed even in regions where rainfall has not increased and, in some cases, has declined. However, this does not mean water constraints disappear. Reduced transpiration can alter local humidity, surface temperatures, and soil moisture dynamics in ways that feed back into the climate system. A plant conserving water at the leaf level may still suffer if heat stress, drought duration, or soil degradation exceeds its tolerance. CO₂ does not remove environmental limits, it shifts them. Nutrients impose another hard boundary. Carbon is only one ingredient in plant growth. Nitrogen, phosphorus, potassium, and micronutrients must be present in sufficient quantities for biomass increases to continue. In nutrient-poor soils, common in large parts of the tropics, plants may initially respond to rising CO₂ and then plateau as other constraints dominate. Long-term fertilization experiments in natural ecosystems consistently show diminishing returns unless nutrients are also available. This is where simplified narratives break down. The same physiological mechanism that enhances growth in one ecosystem may do very little in another. The science does not support a universal outcome. It supports conditional responses shaped by local context. Rising CO₂ creates potential for increased growth, but whether that potential is realized depends on water, nutrients, temperature, and species composition. What emerges from this is not a story of deception or suppression, but one of selective emphasis. Media portrayals often focus on the climatic risks of CO₂ while treating its biological effects as minor or irrelevant. Yet at the level of plant physiology, those effects are neither small nor speculative. They are mechanistic, repeatable, and observable at scale. In the next segment, we will zoom out from the leaf to the planet, examining what satellites actually measure when scientists talk about global greening, how robust those measurements are, and where the signal is strongest and weakest across Earth's land surface. When scientists talk about the Earth greening, they are not relying on anecdotes, isolated field studies, or selective photography. They are referring to decades of continuous satellite measurements that track vegetation cover across nearly every land surface on the planet. These measurements do not infer plant growth indirectly. They detect it through changes in how Earth reflects light. Vegetation interacts with sunlight in a distinctive way. Healthy green leaves absorb most visible red light for photosynthesis while strongly reflecting near-infrared light. Satellites exploit this contrast using indices such as the Normalized Difference Vegetation Index, or NDVI, which acts as a proxy for leaf area, canopy density, and overall photosynthetic activity. While NDVI is not a direct measure of biomass, long-term changes in this signal correlate strongly with shifts in vegetation cover and productivity. Since the early 1980s, multiple satellite platforms, built by different agencies, using different instruments, have recorded a persistent increase in average NDVI across large portions of the globe. This trend is not subtle. By the 2010s, researchers estimated that global leaf area had increased by roughly 5 to 15% compared to early satellite-era baselines. In absolute terms, this represents an expansion of vegetation equivalent to adding a continent-sized area of green leaves. What makes this finding particularly robust is its convergence. Independent research teams, using separate datasets and analytical methods, consistently detect the same greening signal. The pattern appears in data from NASA, ESA, and other international programs. Instrument drift, calibration errors, and methodological artifacts have been examined repeatedly and cannot plausibly explain the magnitude or geographic coherence of the observed changes. The greening, however, is not uniform. The strongest signals appear in high-latitude regions, particularly across parts of Canada, Scandinavia, and Siberia, where warming temperatures extend growing seasons and reduce cold stress. Semi-arid regions, including parts of Australia, the Sahel, and western China, also show notable increases in vegetation cover, often linked to improved water use efficiency under higher CO₂ concentrations. Meanwhile, some tropical regions show weaker trends or even localized declines, especially where deforestation or land degradation dominates. Crucially, researchers do not treat this greening as evidence that ecosystems are improving in a moral or ecological sense. Greening simply means more leaves or longer periods of active growth. It does not automatically imply greater biodiversity, healthier soils, or long-term stability. A monoculture plantation and a diverse forest can produce similar satellite signals while representing very different ecological realities. Attribution studies attempt to disentangle the drivers behind this greening. By combining satellite observations with climate data, land use records, and ecosystem models, scientists estimate the relative contributions of rising CO₂, temperature changes, nitrogen deposition, and human land management. Across many studies, rising atmospheric CO₂ consistently emerges as a dominant driver, often accounting for a third or more of the observed greening signal at the global scale. In some regions, particularly those not undergoing major land use change, its contribution is even larger. This is where public perception often diverges from the evidence. Media portrayals frequently emphasize vegetation stress, forest dieback, and desertification as global defaults. These phenomena are real and important, but they coexist with, and in some cases are outweighed by, simultaneous increases in plant growth elsewhere. The planet is not responding in a single direction. It is responding in multiple, sometimes contradictory ways at once. Satellite data also reveal an important nuance. Greening trends can mask underlying vulnerabilities. Increased leaf area may increase water demand during heat waves, making ecosystems more sensitive to drought extremes even as average productivity rises. In some forests, faster growth corresponds with shorter lifespans and weaker wood density, raising questions about long-term carbon storage and resilience. Greening, in other words, does not guarantee durability. Still, the existence of a large-scale greening signal challenges the idea that rising CO₂ has had no positive biological effects at the planetary level. The evidence shows that plants have already responded, measurably and extensively, to the changing atmosphere. Ignoring that response creates an incomplete picture of Earth system dynamics. In the next segment, we will examine how climate models incorporate CO₂ fertilization, where they succeed, where they struggle, and how uncertainty in biological feedbacks influences projections of future climate and ecosystem behavior. Climate and ecosystem models sit at the uneasy boundary between what we can measure and what we must estimate. They are not crystal balls, nor are they ideological tools by default. They are structured attempts to represent complex physical and biological systems using equations, assumptions, and parameters constrained by available data. When it comes to vegetation and rising CO₂, models both illuminate important dynamics and expose the limits of our current understanding. At their core, Earth system models simulate how energy, carbon, water, and nutrients move through the atmosphere, land, and oceans. Vegetation is not treated as scenery, it is an active participant. Plants absorb carbon, release water vapor, alter surface reflectivity, and influence atmospheric circulation. As a result, changes in plant growth feed back into climate itself, sometimes amplifying trends and sometimes damping them. CO₂ fertilization is built into virtually all modern land surface and ecosystem models. Photosynthesis responds directly to atmospheric CO₂ concentrations, and models reproduce the basic physiological mechanisms observed in laboratory and field experiments. When CO₂ rises, simulated plants grow faster and lose less water, at least initially. In this sense, models broadly agree with satellite observations that rising CO₂ should produce some degree of greening. Where models diverge is in how long these benefits persist and how strongly they interact with other constraints. Nutrient availability is one of the largest sources of uncertainty. Many early generation models assumed unlimited nitrogen and phosphorus, allowing CO₂ fertilization to operate freely. These models often predicted strong and sustained increases in plant productivity, particularly in forests. Later models, which explicitly represent nutrient cycling, tend to show more modest and transient responses as nutrient limitations gradually suppress further gains. Water stress introduces another layer of complexity. Higher CO₂ can improve water use efficiency, but rising temperatures increase evaporative demand, sometimes faster than plants can compensate. In some regions, models project that CO₂ fertilization delays drought stress rather than preventing it. Growth improves in average years, but collapses more sharply during extreme events. This produces ecosystems that look healthier in mean conditions, yet are more vulnerable at the margins. Feedbacks further complicate interpretation. Increased vegetation cover can cool the land surface through enhanced evapotranspiration, but it can also darken the surface, absorbing more solar radiation, especially in high-latitude regions where snow cover is reduced. In boreal forests, this albedo effect can partially offset the cooling benefit of additional carbon uptake. Models must balance these competing influences, and small differences in assumptions can produce divergent outcomes. This uncertainty is often lost in public discussions. Model projections are frequently presented as precise trajectories rather than ranges bounded by assumptions. The role of CO₂ fertilization, in particular, is sometimes downplayed because it introduces outcomes that complicate simple narratives of linear harm. Yet among modelers, the fertilization effect is not controversial. Its magnitude, persistence, and interaction with other stressors are. It is also important to recognize that models are regularly tuned and evaluated against observations. The satellite-detected greening trend forced many modeling groups to revise earlier assumptions about vegetation response. Models that failed to reproduce observed greening were not treated as neutral, they were flagged as incomplete. This iterative process reflects how science actually progresses, not through consensus statements, but through friction between prediction and measurement. Still, models cannot resolve everything. They struggle with local soil processes, species-specific responses, microbial feedbacks, and disturbance regimes such as fire, pests, and land management. A forest in a model is often a functional type rather than a real assemblage of species with different tolerances and lifespans. As a result, model greening may capture the direction of change without capturing its ecological texture. From a public perception standpoint, this creates a subtle problem. When models project future risks, heat waves, droughts, ecosystem stress, the presence of CO₂-driven greening can appear to weaken the message if not handled carefully. Rather than integrate both effects, communication often emphasizes one and omits the other. The result is not necessarily misinformation, but an incomplete representation of system behavior. The scientific reality is more conditional. Rising CO₂ has already altered vegetation at the global scale, and models that ignore this effect perform poorly against observations. At the same time, fertilization does not guarantee long-term stability, resilience, or ecological health. It is a modifier, not a solution. In the next segment, we will turn from models to controlled experiments in the real world, specifically long-term CO₂ enrichment studies, and examine what they reveal about growth limits, nutrient bottlenecks, and why early gains do not always persist. While satellites show what is happening and models explore what might happen, controlled experiments attempt to isolate why it happens. To understand how plants and ecosystems truly respond to rising carbon dioxide, researchers have spent decades running long-term enrichment experiments in fields, forests, and grasslands. These are not laboratory curiosities. They are among the most expensive and technically demanding ecological studies ever conducted. The most influential of these experiments are known as free-air CO₂ enrichment, or FACE studies. Rather than enclosing plants in chambers, FACE systems release CO₂ into open-air plots, allowing vegetation to grow under elevated concentrations while remaining exposed to natural weather, soil conditions, pests, and competition. This design avoids many of the artifacts that plagued early greenhouse experiments and offers a closer approximation to real ecosystems. Across a wide range of FACE experiments, the initial results are strikingly consistent. When CO₂ levels are raised, typically to concentrations expected later in this century, photosynthesis increases almost immediately in C3 plants. Leaf area expands, biomass accumulation accelerates, and water use efficiency improves. Crops like wheat and rice often show yield increases under otherwise favorable conditions, while young trees exhibit faster early growth. However, these gains rarely scale linearly over time. In many experiments, the strongest growth response occurs in the first several years and then gradually diminishes. This pattern does not imply that CO₂ fertilization disappears. It implies that other constraints assert themselves. Nitrogen availability is the most common limiter. As plants grow faster, They deplete accessible nitrogen in the soil, reducing the ability of photosynthesis gains to translate into continued biomass accumulation. In forest face experiments, this effect becomes especially clear. Trees may show enhanced leaf growth and increased carbon uptake, but without sufficient nitrogen or phosphorus, that carbon is often allocated to short-lived tissues rather than long-term structural wood. In some cases, trees grow faster but do not become significantly larger or more durable. Faster growth can even correlate with lower wood density, raising questions about long-term carbon storage and forest stability. Grassland experiments reveal similar trade-offs. Elevated CO₂ often favors certain species over others, subtly reshaping plant communities. Some fast-growing grasses or forbs outcompete neighbors, reducing biodiversity even as total biomass increases. This highlights an important distinction. Ecosystem productivity can rise, while ecosystem complexity declines. Water-limited systems offer a partial exception. In semi-arid environments, the water-saving effects of elevated CO₂ can sustain productivity gains longer than in nutrient-rich but water-stressed systems. Here, reduced stomatal opening meaningfully alters soil moisture dynamics, sometimes allowing plants to survive dry periods that would previously have caused dieback. This mechanism aligns closely with satellite-observed greening in arid and semi-arid regions. Yet even in these systems, benefits have limits. Extreme droughts, heat waves, or shifts in rainfall timing can overwhelm CO₂-driven efficiencies. Experiments repeatedly show that elevated CO₂ does not immunize ecosystems against climate extremes. It changes the baseline, but does not eliminate risk. One of the most important insights from long-term enrichment studies is that ecosystems adapt. Soil microbial communities shift, nutrient cycling pathways adjust, and plant allocation strategies evolve over time. The ecosystem that exists after a decade of elevated CO₂ is not the same system that existed at the start of the experiment. This dynamic adaptation is difficult to capture in models and easy to overlook in public discussions. From a communication standpoint, FACE results complicate simple narratives. They confirm that rising CO₂ directly stimulates plant growth and water efficiency, validating the physiological mechanisms observed in labs and inferred from satellites. At the same time, they demonstrate that these benefits are neither unlimited nor uniformly beneficial. Productivity gains often come with trade-offs in nutrient balance, species composition, and long-term resilience. The mistake is not acknowledging CO₂ fertilization. It is treating it as either a cure-all or a trivial footnote. The experiments show that CO₂ is a real driver, operating within real constraints. Any serious assessment of global vegetation change must account for both. In the next segment, we will examine how agriculture fits into this picture, whether crops respond differently than wild ecosystems, how yield gains compare to stress losses, and why food production trends further complicate public perception of a warming, higher CO₂ world. When the subject turns to agriculture, discussions of CO₂ and climate tend to sharpen quickly. Food sits at the intersection of biology, economics, and risk, and the stakes feel immediate. Yet the agricultural response to rising CO₂ is often portrayed in stark, simplified terms that miss how crops actually behave under changing atmospheric conditions. From a physiological standpoint, most major food crops are C3 plants. Wheat, rice, soybeans, barley, potatoes, and many legumes respond to elevated CO₂ in ways that closely mirror experimental results seen in natural ecosystems. Photosynthesis becomes more efficient, water use efficiency improves, and under controlled conditions, yields often increase. These responses have been observed repeatedly in greenhouse studies and open-air enrichment experiments. In FACE experiments focused on crops, yield increases under elevated CO₂ typically range from modest to significant, depending on water and nutrient availability. Wheat and rice often show increases on the order of several percent to low double digits when other stresses are minimized. In water-limited environments, the water-saving effect of CO₂ can be as important as the fertilization effect itself, allowing crops to maintain productivity during dry periods that would otherwise reduce yields. However, agriculture does not operate in isolation from climate. Heat stress, extreme weather, pests, and diseases interact with CO₂ responses in complex ways. High temperatures during critical growth phases, such as flowering, can sharply reduce yields, even if average photosynthesis improves. Elevated CO₂ does not prevent heat-induced sterility in crops like wheat or rice, nor does it fully compensate for damage caused by droughts that exceed historical norms. This leads to an important but often overlooked distinction. Rising CO₂ tends to increase potential yield, the amount a crop could produce under favorable conditions, while climate variability increasingly determines whether that potential is realized. In some regions, technological improvements, irrigation, and crop breeding have allowed farmers to capture much of the CO₂-driven benefit. In others, climate stressors dominate, masking or overwhelming fertilization gains. Nutritional quality adds another layer of complexity. Elevated CO₂ often increases carbohydrate content faster than increases mineral and protein content. As a result, crops grown under higher CO₂ can exhibit reduced concentrations of zinc, iron, and protein per unit mass. This does not mean food becomes nutritionally useless, but does mean that higher yields do not translate one-to-one into improved nutrition, especially in regions already vulnerable to micronutrient deficiencies. Globally, agricultural production has continued to rise over recent decades, even as atmospheric CO₂ and temperatures have increased. This trend reflects a combination of factors: improved genetics, fertilizer use, mechanization, pest control, and expanded irrigation. CO₂ fertilization is part of this picture, but it is not the dominant driver. At the same time, claims that climate change has already caused a collapse in global food production are not supported by the data. Production has grown, though unevenly, and with increasing exposure to climate risk. Public perception often struggles with this nuance. Media narratives may emphasize vulnerability and impending crisis, while others point to rising yields as proof that climate impacts are exaggerated. Both interpretations oversimplify. The evidence suggests that CO₂ has made crops more efficient and productive under many conditions, while climate variability has made outcomes less predictable and more regionally unequal. Another subtle effect emerges at scale. Improved water use efficiency under elevated CO₂ can reduce crop transpiration, altering local hydrology. In heavily irrigated regions, this may slightly reduce water demand. In rain-fed systems, it can influence soil moisture retention and microclimates. These feedbacks are rarely visible to farmers in the short term, but matter in long-term water planning. From a scientific standpoint, agriculture demonstrates the core theme of this series more clearly than any other domain. Rising CO₂ produces real, measurable biological benefits, but those benefits operate within systems shaped by multiple constraints and human decisions. Ignoring CO₂ fertilization leads to overly pessimistic projections. Overstating it leads to complacency. In the next segment, we will broaden the focus again and examine how rising CO₂ interacts with forests and carbon storage, whether greening translates into long-term carbon sinks, or whether faster growth creates more fragile ecosystems. Forests occupy a special place in the climate conversation because they are not just responders to atmospheric change, they are major regulators of it. Through photosynthesis, forests absorb vast quantities of carbon dioxide, storing it in wood, roots, and soils over timescales that range from decades to centuries. If rising CO₂ causes forests to grow faster, the implication seems straightforward. More growth should mean more carbon removed from the atmosphere. In practice, the story is more complicated. Observational data and long-term forest inventories show that many forests have indeed increased their rates of carbon uptake over the past several decades. Tree growth rates have accelerated in parts of North America, Europe, and the boreal regions, consistent with both CO₂ fertilization and longer growing seasons. Satellite data reinforce this picture, showing expanding canopy cover and increased photosynthetic activity across large forested areas. Yet faster growth does not automatically translate into long-term carbon storage. Carbon remains sequestered only as long as the biomass that contains it persists. If trees grow quickly but die younger, or if forests become more vulnerable to disturbance, the net climate benefit may be smaller than raw productivity numbers suggest. One concern emerging from both experiments and forest monitoring is wood density. Elevated CO₂ often promotes rapid growth, but faster growth can produce less dense wood, particularly when nutrients are limiting. Less dense wood stores less carbon per unit volume and may be more susceptible to mechanical failure, pests, and decay. This does not mean that CO₂ fertilization weakens forests universally, but it does introduce trade-offs between growth rate and structural resilience. Disturbance regimes further complicate the picture. Fire, insect outbreaks, storms, and disease play decisive roles in determining how long forest carbon remains stored. In some regions, increased vegetation growth provides more fuel for wildfires, increasing the likelihood of large carbon releases during extreme events. In others, warmer temperatures allow pests to expand into previously inhospitable areas, stressing trees that are growing faster but not necessarily stronger. Soils represent another major uncertainty. A large fraction of forest carbon resides below ground, stored in roots and organic matter. Elevated CO₂ can increase root growth and carbon inputs to soil, but can also stimulate microbial activity that accelerates decomposition. Whether soils gain or lose carbon under rising CO₂ depends on temperature, moisture, and microbial dynamics, factors that vary widely across regions and are difficult to predict. Despite these uncertainties, most lines of evidence suggest that terrestrial ecosystems, including forests, have acted as net carbon sinks over recent decades, absorbing a significant fraction of human CO₂ emissions. This sink is partly driven by CO₂ fertilization and partly by regrowth on previously cleared land. However, its future persistence is not guaranteed. As forests age, saturate, or experience more frequent disturbances, sink strength may weaken even if greening continues. Public perception often frames forests either as climate saviors or as imminent victims. Both framings obscure the middle ground revealed by data. Forests are responding dynamically to rising CO₂ and climate change, gaining productivity in many areas while facing increasing stress in others. Their role as carbon sinks is real but conditional, influenced by management practices, disturbance control, and regional climate trajectories. From a scientific standpoint, the key insight is that greening does not equal permanence. A greener planet today does not automatically mean a more stable climate tomorrow. Carbon storage depends not only on how much grows, but on how long it stays locked away. In the next segment, we will turn toward the other side of the equation, where greening does not occur, where vegetation declines or fails to respond to rising CO₂, and what these exceptions tell us about the limits of biological buffering. The existence of widespread greening does not mean that all ecosystems are benefiting from rising CO₂, nor does it mean that negative impacts are rare or exaggerated. In many regions, vegetation shows little response to higher CO₂, and in others it is declining outright. Understanding where greening fails is essential for separating genuine biological signals from overgeneralized conclusions. Temperature is one of the most decisive constraints. Photosynthesis operates within a finite thermal window. Beyond certain thresholds, enzymatic efficiency declines, respiration rates increase, and net carbon gain falls. Even if CO₂ is abundant, excessive heat can overwhelm the photosynthetic machinery. In tropical regions, where temperatures already approach optimal limits, additional warming can reduce productivity rather than enhance it. This helps explain why some equatorial forests show weak greening signals or signs of stress despite rising CO₂. Drought presents a related but distinct challenge. While elevated CO₂ improves water use efficiency, it does not create water where none exists. Extended droughts, particularly those accompanied by high temperatures, can drive widespread vegetation mortality. In such conditions, stomatal closure limits CO₂ uptake regardless of atmospheric concentration, halting photosynthesis and forcing plants into survival mode. Repeated drought stress can push ecosystems past recovery thresholds, leading to long-term degradation. These failures are visible from space. Satellite data reveal regions where vegetation indices stagnate or decline, often coinciding with areas experiencing increased heat extremes, reduced rainfall, or land degradation. Parts of the Amazon, for example, show signs of stress during severe drought years, with reduced photosynthetic activity and delayed recovery. Similarly, Mediterranean and subtropical regions exhibit growing sensitivity to heat and water scarcity, offsetting potential CO₂ benefits. Land use change frequently dominates biological signals. Deforestation, urban expansion, overgrazing, and soil erosion can suppress vegetation regardless of atmospheric conditions. In such regions, the absence of greening is not a failure of CO₂ fertilization, but a consequence of direct human disturbance. This distinction matters because it affects how observations are interpreted and communicated. Another critical limit arises from ecosystem structure itself. Some systems are constrained not by carbon or water, but by disturbance regimes that reset vegetation before long-term gains can accumulate. Fire-prone landscapes, for example, may experience increased biomass under elevated CO₂, only to lose it during more frequent or intense fires. The net effect over time may be neutral or negative, even though growth rates between disturbances increase. Physiological acclimation also plays a role. Plants do not respond indefinitely to elevated CO₂. Over time, photosynthetic machinery can downregulate, particularly when nutrient availability cannot keep pace with increased carbon assimilation. This reduces the long-term fertilization effect and contributes to the flattening of greening trends observed in some long-term datasets. These exceptions are often emphasized in media coverage, sometimes to the exclusion of broader patterns. Heat waves, drought-induced dieback, and forest loss make compelling stories, and they matter. But when presented as representative of global behavior, they can distort public perception. The scientific picture is not one of uniform decline or uniform improvement, but of heterogeneous responses shaped by local thresholds. Importantly, regions where greening fails often coincide with areas of high human vulnerability. Agricultural systems in marginal climates, water-stressed cities, and ecosystems already near physiological limits are less able to capitalize on CO₂-driven efficiencies. This reinforces a central theme. CO₂ fertilization is context-dependent. It operates where constraints allow it, and fails where limits dominate. In the next segment, we will step back from biophysics and examine how these scientific nuances are communicated or lost in public discourse. We will explore why media narratives often diverge from research findings, how framing choices influence perception, and what gets simplified or omitted when complexity meets attention economics. Scientific understanding advances slowly through accumulation, correction, and conditional conclusions. Public understanding, by contrast, is shaped by narratives, compressed explanations designed to be quickly understood, emotionally resonant, and repeatable. When these two modes intersect, something is often lost. The story of CO₂ and global vegetation illustrates this tension with unusual clarity. Most media coverage of climate-related topics operates under severe constraints. Headlines must compete for attention, stories must fit limited space, and complex interactions must be rendered into simple cause and effect relationships. In this environment, ambiguity becomes a liability. A message that says CO₂ both stimulates plant growth and increases certain risks depending on context is harder to convey than one that frames CO₂ as uniformly harmful or uniformly beneficial. As a result, CO₂ fertilization tends to be treated as either negligible or suspicious. When greening is mentioned, it is often framed as temporary, misleading, or irrelevant to long-term climate outcomes. While these caveats have scientific merit, their prominence can create the impression that vegetation responses are minor side effects rather than central components of Earth system dynamics. This is not how the subject is treated in the research literature, where plant responses are integral to carbon cycle modeling and climate projections. Another factor shaping perception is the distinction between global averages and local impacts. Media stories often focus on dramatic regional declines, forest dieback, crop failures, drought-stricken landscapes, because they are visually compelling and socially relevant. These events are real and deserve attention, but they coexist with quieter, less visible trends of increased growth elsewhere. Without explicit context, audiences may assume that localized degradation reflects global behavior. Scientific papers, by contrast, are explicit about scale. A single drought does not negate a multi-decadal greening trend, just as a global average does not eliminate regional suffering. When this distinction is lost in translation, public understanding tilts toward absolutism. There is also a cultural factor at play. CO₂ fertilization challenges intuitive moral framing. It introduces a benefit associated with a substance widely labeled as a problem. Communicators may worry, sometimes legitimately, that emphasizing biological benefits could be misinterpreted as minimizing climate risks or discouraging mitigation efforts. In response, coverage may emphasize harms while downplaying countervailing effects, even when both are present in the data. This dynamic does not require conspiracy or intent. It emerges naturally from incentive structures. Journalists respond to audience attention, institutions respond to political and funding environments, scientists respond to peer norms and caution. Over time, certain aspects of a science receive repeated amplification, while others fade into technical appendices and specialist journals. The result is an incomplete mental model for the public. Many people believe that rising CO₂ has already caused widespread and uniform vegetation decline when the empirical record shows a more mixed response. Others believe that greening invalidates climate concerns altogether, overlooking constraints, trade-offs, and vulnerabilities. Both positions arise from partial information. What is rarely communicated clearly is that CO₂ fertilization and climate stress are not opposing explanations, they operate simultaneously. The biosphere can become greener on average while becoming more fragile at the margins. Productivity can increase while resilience decreases. These are not contradictions, they are layered outcomes of a complex system adjusting to new boundary conditions. Public perception tends to collapse these layers into a single storyline. Science does not. The gap between the two is not primarily about facts, but about framing, emphasis, and tolerance for nuance. In the final segment, we will draw these threads together, reviewing what the evidence actually supports, where uncertainty remains, and how a more accurate public understanding of CO₂ in vegetation could improve both scientific literacy and policy discussions without resorting to simplification or denial. After decades of observation, experimentation, and modeling, the scientific picture of rising CO₂ and global vegetation is neither comforting nor catastrophic. It is conditional, uneven, and deeply context-dependent. Yet within that complexity, several conclusions are remarkably stable. First, rising atmospheric CO₂ does stimulate plant growth under many conditions. This is not speculative and not controversial within plant science. The physiological mechanisms are well understood, repeatedly demonstrated in experiments, and visible at planetary scale through satellite observations. Large portions of Earth's land surface have become greener over recent decades, and CO₂ fertilization is a significant contributor to that trend. Second, this greening does not operate in isolation. Water availability, nutrient supply, temperature extremes, land management, and disturbance regimes all shape how much of the CO₂-driven potential is realized. Where constraints are mild, vegetation often responds strongly. Where constraints dominate, through heat, drought, poor soils, or direct human disturbance, CO₂ provides little protection. Third, increased growth does not guarantee long-term ecological stability. Faster-growing plants may allocate carbon differently, alter species composition, and interact with soils and microbes in ways that reduce durability. Forests can absorb more carbon while becoming more vulnerable to fire, pests, or mortality. Productivity gains and resilience are not the same variable, even though they are often conflated in public discussions. Fourth, models that incorporate CO₂ fertilization generally perform better against real-world observations than those that do not. At the same time, models struggle to represent nutrient limitations, disturbance dynamics, and ecosystem adaptation over long timescales. This means future projections carry genuine uncertainty, not because the science is weak, but because biological systems are adaptive and historically contingent. Fifth, the public narrative surrounding CO₂ and vegetation is often incomplete. Emphasis on harm without acknowledgement of biological response creates a distorted picture, just as emphasis on greening without attention to limits invites complacency. The scientific literature does not support either extreme. It supports a world in which the biosphere is actively responding to rising CO₂, sometimes beneficially, sometimes insufficiently, and sometimes in ways that create new vulnerabilities. Perhaps the most important insight is this: rising CO₂ is not a passive background change. It is an active driver reshaping plant physiology, ecosystem structure, and land-atmosphere feedbacks right now. Any serious attempt to understand climate, food security, or carbon cycling must account for these effects, not as footnotes, but as core processes. Recognizing CO₂ fertilization does not require denying climate risks. It requires accepting that Earth systems respond in more than one direction at once. The challenge moving forward is not choosing between optimism and alarm, but developing frameworks that can accommodate both adaptation and stress without collapsing into simplification. In that sense, the reality differs from how the subject is often depicted, not because the science contradicts concern, but because concern alone cannot capture the full behavior of a living planet adjusting to new conditions. And that ultimately is the InSlowSpective view: slower, broader, less certain, but closer to how the world actually works.